Yes.
Good morning, one and all. I am Mrs. Sonali Labdar, Assistant Professor, Dr. Y. Patil, Institute of Pharmaceutical Sciences and Research, Pimpri, Pune. Feel privileged to welcome you all on behalf of Training and Placement Cell, Dr. D. Y. Patil, Institute of Pharmaceutical Sciences and Research, Pimpri, Pune, to one day national level webinar on cytometry in pharmacy, basic principle and its application. Well, Jane William R. once said, the aim of education is the knowledge, not of fact, but of value. It is on the occasion like this, we get opportunities to test our knowledge and understanding. We look forward to get an exposure about what the best of brains think about this very dynamic issue of FCP. I request all participants to keep your videos off and volume mute. You can post your questions in chat box, which will be discussed at the end of the session. Feedback link will be provided at the end of question answer session. Now, I would take opportunity to introduce our today's speaker, Dr. Nishant Vyas. Sir presently is Director at Logical Biosciences, Pune. Sir has completed his doctorate in human immunology, mainly working on single-cell cloning of regulatory T cells. Sir has been awarded several CSIR Junior Research Fellowships. Sir has received call for prestigious Shaina Prasad Mukherjee Award 2 times. Sir has in his credit several scientific achievements and technical achievements in the field of biotechnology and microbiology. Sir has analyzed 300 anti-cancer molecules on 3 breast cancer cell lines in a day with flow cytometry. Sir has 10 years of experience in multicolor flow cytometry on machine platforms like FSC, LSR, Canto, etc. Sir research interest is mainly in creating tumor-specific effector T-cell clones while rebooting T-cell exhaustion from oral and breast cancer origin. Characterization of cancer stem cells and cancer stem cells, aging induction in cancer stem cells and analysis. Now, without taking more time, I request Dr. Nishant Vyas to start with the session. Please, sir.
Thank you, ma'am. Thank you very much. First of all, I'm grateful you guys from Dr. D. Y. Patil College of Pharmacy to provide me this opportunity to talk on flow cytometry, and the topic is flow cytometry in pharmacy, basic principle and applications. So I know that students and researchers in the pharmaceutical sciences, they are working a lot in the field of drug discovery, drug design, and the effect of drugs on human system and mouse models, rat models, and so many things. But I would like to say that flow cytometry can make so many things very easy in your field, and I have kept my presentation as student orientation specific as possible, as simple as possible, because I personally believe that we should discuss the science which we can explain to students in the simplest manner possible. So we'll go through the principle of flow cytometry, how the system is working, how the technology is working, and then we will see some very interesting applications. And in between, I will be giving 2 or 3 my advices to the students, which can help them in changing their perspective towards biology as subject or perspective towards science as subject. This is the game about how you think towards biology, how you address relevant research questions, how you apply the technology. Then you can make good career, as well as then you can make your life very easy with the help of tools and techniques of biology. So let's start simply with the basic principle. So if somebody is asking me, what do you mean by cytometry? Then I will be giving the answer that we are going to measure physical and chemical properties of single cell, or you can say one cell at a time. So if we are having a method where we can capture single cell at a time, and we can measure the physical property of that particular cell or chemical property of that particular cell, then that technology will be falling under the cytometry. And if I just add a word flow in front of cytometry, then it becomes flow cytometry. We are doing the same thing. We are measuring the physical property. We are measuring the chemical properties of a single cell in an instrument which is using flow of a fluid. A fluid stream is been utilized to understand the physical property and chemical property of that particular cell. And then we start calling it as what? Flow cytometry. So from the term flow cytometry, now the things are very simple. We are going to have one instrument which will create a stream of fluid to capture one cell at a time, a single cell at a time. And the instrument will be giving the readings. We will be utilizing those readings to understand the physical and chemical properties of that particular cell. And our flow cytometry is done. So as I said, what our instrument will be doing, we'll go later on that. But before that, I always tell the people that to learn the technology or to learn a technique or to learn an experiment in a better way, the best way to do that is you design one experiment. So let's design a hypothetical experiment and then we run that experiment into the flow cytometer and let's see what will happen. So first of all, we can take a tissue or we can take cells, okay? And from this tissue and from these cells, we can prepare a single cell suspension. What do you mean by single cell suspension? Means a buffer containing your cells which you want to analyze, but they should not be in form of clumps. They should not be in form of aggregates. They should not be in form of tissue. They should be in form of single cell. So we can take a tissue and we can treat the tissue with enzymes like collagenase, elastase, displase. These enzymes can break cell to cell connections and we can have a single cell suspension. If you are working with bacteria, then directly you can use them because they are already in the single cell suspension. If they are growing in liquid cultures, you can use cell lines, you can use cultured cells. The ultimate aim is we have to start the experiment with a tube which is containing a single cell suspension. Now we take this tube and we put that tube into the flow cytometer. Right. Now what will happen? In the next step, what the flow cytometer is doing, here you are putting your cells in the form of single cell suspension and the instrument is creating a flow of fluid to pressurize these cells from both the sides. So ultimately you can achieve a condition where you are creating an array of cells where one cell at a time passing through this array or in form of array. So that is the functionality of instrument. So flow cytometry or flow cytometer is not rocket science or it is not a magical instrument. What the instrument is doing, that you put a single cell suspension into that instrument, the instrument will be creating a high pressurized flow of fluid from both the direction and will be creating a single cell array. Then the single cell will be interacting with laser, which is the part of the instrument. And these interactions, which are of many types. So these interactions will be detected by the detectors and detectors will be giving you the readings and those readings you have to understand. Understanding the reading or understanding the results of experimentation is not the work of instrument itself, but that is the work of the person who is analyzing the data. But you take the cell, you can take the tissue, you can prepare the single cell suspension, and that single cell suspension will be converted into a single cell array where one cell at a time interacting with the laser, and those interactions are recognized or detected by the detectors and they are coming to us in form of readings. So this is the function of what? Flow cytometer. Now if we really want to understand the data of flow cytometry and we really want to apply some good applications of flow cytometry, then we have to understand what kind of interaction our single cell is having with this laser. And if we are able to understand these interactions, then it is very simple to understand the principle of flow cytometry, as well as it is very simple to design so many different types of experiments, to design so many different types of applications in your field by utilizing this very advanced and very beautiful technology. So now I'm going to explain what kind of interaction can be there between this laser beam or this beam of photons and our single cell. So imagine this is our single cell, and this laser is putting a light beam on this single cell. And I think in class 11th or 12th, everybody of us have understood that if a light is interacting with a solid object, then there can be scattering of light. So similar thing will be happening here. This light is coming from the laser and this will be scattered by our single cell. Now what happens? That scattering of light can be because of edges of the cell. And if the scattering is happening because of edges of our single cell, then the angle of scattering is between 0.5 degree to 5 degree. So the light is coming on, you can say 0 degree angle, and suddenly it has scattered to 0.5 degree to 5 degree angle. It is changing its path, and the change in path is between 0.5 degree to 5 degrees. Then it is happening because the light is getting scattered from the edges of the cell. And this scattering, we call it as forward scatter or FSC. This is the first parameter which is given by flow cytometer. Now, forward scatter is actually giving you the information about size of the cell. So if you are having bigger cell, then there will be higher value of forward scatter or higher value of scattering caused because of size of cell. Now, higher the value of FSC means you are having bigger cell. Lower the value of FSC or forward scatter, you are having a smaller cell. So by using the values of FSC and by using this first parameter, forward scatter, we are understanding the size of the cell. Now, the light can enter inside the cell, and inside the cell, what we are having biomolecules, we are having cellular organelles, we are having nucleus, we are having endoplasmic reticulum, we are having mitochondria. So all because of these granules or these bodies, the light can scatter, and the angle of that scattering of light comes between 50 degrees to 150 degrees. And we call this parameter as SSC or side scatter. So the SSC is giving you information about the internal granularity of cell. Internal granularity of cell means more granules are present, then there will be higher value of SSC or higher value of side scatter, and less granules are present, then it will be lesser value of what? Side scatter. So we are getting two physical parameters. Number one, size, which we are calling as FSC, and second, granularity of cell, which we are calling as SSC or side scatter. The third parameter which you can measure with these interactions, it is possible that your cell is having some kind of fluorescent factor present in it. Whether this fluorescent factor is natural or it is artificial added by you, like some kind of fluorescent dyes, some kind of fluorescent labeled antibodies, some kind of fluorescent protein, some kind of recombinant proteins. So the fluorescence factor can be a natural one or can be artificial one, that doesn't matter. But these fluorescence factors will be absorbing a particular wavelength and they will be emitting a particular wavelength, and that emission of wavelength again we can detect by what? Our detectors. So the flow cytometry or the instrument is giving you three information about a single cell or about your population of cell. Number one, forward scatter of the size. Number two, side scatter of the cell, that is granularity of the cell, and presence and absence of a fluorescent factor or fluorescent protein or fluorescent biomolecule inside your cell or fluorescent artificial molecule inside your cell. Size, granularity, and fluorescence. These three informations you are actually getting from your flow cytometer. And that is the basic principle of flow cytometer. So I can summarize. Forward scatter is giving you the information about size of cell. Side scatter is giving you the information about internal granularity of cell. And there can be fluorescent signals coming out of the cell. The cell can have inherent fluorescent proteins. Sometimes happen in algae, you know, that brown algae, blue-green algae, and green algae, they create different type of inherent fluorescent proteins. Then the fluorescent proteins with the help of genetic engineering, like GFP, green fluorescent protein, red fluorescent protein, RFP, yellow fluorescent protein, YFP, cyan fluorescent protein, CFP, we can do that. Otherwise, we can use antibodies, which are fluorescent tagged antibodies. And these antibodies are binding to a target protein. And those target proteins, with the help of these antibodies, are giving us what? Fluorescence. And we can use fluorescent dyes for the staining of parts of a particular cell, like DNA, like nucleus, like some kind of protein. So size, granularity, and fluorescence. We'll be playing with these factors, and I will be explaining some amazing experimentation which can be done by flow cytometry with your drugs. So whatever I am going to discuss now, different application, every time you keep in your mind, if you're working on a particular drug molecule, and then you can have a question in your mind that how I can utilize this technology, these parameters, to understand the effect of my drug or my herbal extract or my interest molecule or my molecule of interest, how I can apply this. Then this lecture or this discussion will be very useful for you. So I hope I'm not boring you and I'm not going very fast. So there are some fluorescent dyes which can be utilized. There are some names which you don't have to remember, obviously. FITC, that is coming into green, PE, that is coming into red, then APC, that is coming into blue. And the combination of these colors, like APC 647, LXR 488, then some fluorescent proteins are there, as I said, CFP, GFP, YFP, RFP. They are having some absorbance, they are having some emittance, they are having a signal of emission. But overall, size, granularity, and fluorescence. Now let's start doing the application. So we start with one very simple application that you have injected. So I hope there are pharmacology students here or people who are working in the field of pharmacology here. So imagine you have added or you have injected a drug intravenously and to see whether that particular drug can analyze or can do the immunomodulation in the rat, mouse, or human or whatever. So you want to check the immunomodulatory property and you want to count that after injecting that drug or before injecting that drug, what is the number of T cells and B cells, T lymphocytes and B lymphocytes in the body of rat, mouse, human, whatever organism you are taking. So you can do that by flow cytometry. That is not good counting. You can do that by microscopy actually, but that is not the absolute counting. So you don't get absolute numbers, and that's a time-taking process. So can we do that analysis in 5 minutes? The answer is yes. We can analyze the overall system in 5 minutes. How? So after injection, you have taken out the blood, and from blood, by using a technique, a specific centrifugation technique, which is called as Ficol density centrifugation, you have isolated the PBMC. PBMCs are peripheral blood mononuclear cells. Or you can say that you have removed the RBCs, which are not having the nucleus, and you are utilizing the PBMCs, the cells which are having the nucleus. Now, I want to analyze the percentage of T cells and B cells in these PBMCs. So what I do, I know that instrument is not going to help me. Only the knowledge of biological theory or the biological fundamentals will be helping me. So I know very well that T cells, all the T cells in an organism, they express a protein on their surface, which is called as CD3, cluster of differentiation 3. This protein is specific to what? T cells. And another protein is there, CD19, which is expressed mainly by B cells or only by B cells. So I can say that in PBMCs, if I add the antibodies, fluorescent label antibodies against CD3 protein and against CD19 proteins. So the anti-3 will be binding to T cells, and antibodies against CD19 will be binding to what? B cells. So whatever fluorescence is used to label the CD3 antibody, that will be given by only T cells, and whatever the fluorescence which is utilized to label CD19 antibody, that will be given mainly by B cells. So we have added the antibody, we have incubated for 30 minutes, and we are putting the test tube in the flow cytometer. So CD3 is the surface marker of T lymphocytes, and CD19, it is the surface marker of what? B lymphocytes. So you have added, you have mixed the antibodies, you have incubated it, and then you are putting the test tube in your flow cytometer. Now what will happen? Flow cytometer will be creating an array of cells. Lasers will be interacting. Laser will be detecting size, granularity. Fluorescence given by CD3 positive cells means by T cells, and fluorescence given by CD19 cells means B cells. Now we get this kind of result. Now in first shot, it is like, what is this? Kya cheez hai? I mean, it is difficult to understand, but it is very easy to explain if we go step by step manner. So you can see that in the first graph on x-axis, I am having FSC, that is the size of cell, and on y-axis, I am having SSC, that is what? Granularity of cell or site scatter. So here 1 dot is equal to 1 cell. So for example, you have added 1 million cells in your experiment. So you are going to have 1 million dots. So you can see that there are 3 populations, 1 here, then second here, and third here. So the first population is equivalent to the population of lymphocyte, T cells and B cells. The second population is equivalent to monocytes in macrophages. And third population is equivalent to what? Granulocytes, that is eosinophil, basophil, neutrophils and other cells. Now what is happening, that everybody has used the Microsoft Paint. There you can use the selection area and you can select anything. So similarly, by using the software, we can select these cells. And once I select that, instrument automatically gives me the percentage and count how many cells are coming into this gate or in this selected area. So immediately within seconds, I find that in my PBMCs, what is the percentage of lymphocyte? What is the percentage of monocyte? What is the percentage of what? Granulocyte. So this whole experiment is taking 35 minutes and you are having exact count of lymphocyte, monocytes and what? Granulocyte. Amazing thing. Now I want to understand that within lymphocyte, how many are B cells and how many are T cells. So I select this area and then I tell the software that now show me how many cells are CD3 positive. Here it's not 5, it is 3. And how many are CD19. So I again get this graph. Everybody I think has read about the Ramachandran plot or quadrant plots of the graphs. So on x-axis we are having the fluorescence of CD3, and on y-axis we are having the fluorescence of CD19 or the B cells. And the graph is saying the graph can be divided into 4 quadrants. This first quadrant is negative for CD3 and this is also negative for CD19. So these are not T cells as well as these are not B cells, they are something else. Now these cells in this quadrant, they are positive for CD3 but they are negative for CD19. So these are T cells but not the B cells. Now these cells, these dots are positive for CD19 but they are negative for CD3. So they are what? B lymphocytes. And here we don't have any cell which is positive for CD3 as well as for CD19 because we already know that one cell cannot be both B as well as T cell. So the system is calculating the percentage. So you can say in that person after injecting the drug, the T cells are 66.39%, the B cells are 18.11%, and If you increase the drug concentration, you can compare whether this percentage is modulating, whether before injecting the drug and after the treatment of drug, if there are some changes. And this one sample in the instrument after 30 minutes incubation is getting analyzed in 1 minute. Now, in my lab, we are having one flow cytometer, which can analyze 7,200 samples in a day. So you can analyze 7,200 blood samples after creating a huge study to understand whether your drug is able to modulate the count of T lymphocyte and B lymphocyte in the subjects, in the animals, in the patients before and after the treatment. And you can analyze 7,200 samples in 24 hours. So before the flow cytometry, the same thing was taking 3 years to do. After the flow cytometry, the same thing is taking 1 day to complete. It's just, you should be having the imagination and you should be having the knowledge of your biology with you, that T cells will be what? CD3 positive, and B cells will be what? CD19 positive. Very interesting experiment. Quickly, every minute you are getting one data plot. And imagine in your research study, your thesis, or in your paper, you are having 7,200 graphs like this. And you are analyzing a hell lot of data, and you are telling that the effect is increasing, decreasing. So people say that Chyawanprash is immunomodulator, right? But nobody tests that. Nobody checks that. That herbal extract is known to have boosting the immune response. But how to check that? It's very good. I take PBMCs out of my blood. I isolate T cells out of my blood, and I treat the T cells with that particular extract or particular herbal extract or particular drug. After 3 days, I check for the activation markers present on my T cells by the flow cytometry, and I can actually quantify how much the immune system is modulated, how much the immune system is getting activated more, what is the effect of that particular drug. So when we can do everything in real time and everything we can do with the live cells, then why not to increase the throughput of the experiments, as well as why not to utilize the technology? So what you are requiring, actually, you are requiring the simple knowledge of some physics scattering, some chemistry, that what is the fluorescence, where it will be emission, where it will be absorbance, as well as fundamentals of biology, that which cells are expressing which protein, and then you are good. And then you should be having a good imagination to set up the experiment. And yes, you can do that. Right? Interesting experiment. So I always tell the student that here my first advice comes. I always tell the students that even if you are a student of the field of biological sciences, but you should learn the things in an integrated way. Integrated way means you should be able to utilize the principles of physics and chemistry to explain the biology. I generally say there is no subject which is called as biology. Biology is a system where you are combining the knowledge of principles of physics and principles of chemistry to explain the properties of cells. And if you start utilizing your brain in that direction, if you start changing your perspective towards the subject by utilizing this theme, then things will be very easy for you. So even Mother Nature is doing same. It is very interesting to know that when Mother Nature decided that we have to create biological system, and in biological system we have to do polymerization reactions. Polymerization reaction means DNA replication is actually the polymerization of nucleotides. Transcription, it is again mRNA formation, that is polymerization of nucleotides. Then protein synthesis, that is polymerization of amino acids. The glycogen synthesis, starch synthesis, polymerization of carbohydrates. So whenever a polymerization reaction is happening inside the cell or inside a biological system, then Mother Nature has decided that I will not keep the things very complex. I will keep the things very simple. So she has utilized one reaction mechanism for all these polymerization processes, that is a nucleophilic substitution reaction. So if we are very good in understanding the principles of nucleophilic substitution reaction, then automatically it is very easy to understand DNA replication, transcription, translation, polymerization of carbohydrate, whatever polymerization of you are doing in the biomolecular system. And once you are understanding that the whole thing is getting synthesized by utilizing nucleophilic substitution reaction, then it is very easy to design the drug to target that reaction process. I mean, small chemical fundamental can solve big things and can help you in designing a drug. So it is very interesting, and everybody should, I think, should change the perspective towards biology, that more and more amalgamation and more integration of physics, chemistry, and mathematics to understand the field of biology. Right. So we go further and we see one problem which we generally face in the cytometry. This I generally don't discuss in theory because this is the practical part, but yeah, what we can do. So I'm just explaining the problem, but the solution is practical, so I cannot explain it. The problem is, if in single cell suspension, if I'm adding 3 antibodies which are having 3 different fluorescent tags, so what happens, their fluorescence can overlap each other. And sometimes you can get false signals. So you can see I have added 2 antibodies, FITC and PE. FITC labeled antibody and PE labeled antibody. So we face a problem of what? Spectral overlap. Imagine I am adding 1, 2, 3, 4, 5 antibodies. So much overlap of spectrum system, spectral lengths. So it is again difficult to analyze the data. So more and more you design a complex experiment in flow cytometry, more and more it's difficult to explain the data or to analyze the data. So there are different type of instruments. So there is one laser instrument which can analyze only 2 colors, means you add 2 antibodies. We are having a 3 laser instrument which can analyze 14 colors, means simultaneously you can add 14 type of protein specific antibodies. Then there are 4 laser instruments which are having 18 color generation. So which instrument one should buy, that depends upon how much you are able to understand the spectral overlap and how much you are able to analyze the data, as well as how much money you are having in the pocket. Because more the advanced is the instrument, it becomes more and more expensive. So generally when I explain the flow cytometry, people come with this question also, kitne ka aata hai flow cytometry? And how much money you can buy the flow cytometer? So that depends upon what flow cytometer you are buying. So if you are buying a 2 laser, then it costs you around 50 lakhs. And if you are buying 3 laser, then around 90 lakhs to 1 CR, and more than 2 CR, 2.5 CR. So depending upon what are your application, as well as how much you can afford. Yeah, but this problem of spectral overlap can be solved by using some mathematics. So we call that process as compensation. But whenever we'll be having a chance to do it practically, I will be explaining how we can analyze the data of 6 proteins, 7 proteins, 14 proteins at a time. So imagine you can do 7,200 samples in a day, and in each sample you are analyzing 14 parameters. So you multiply 7,200 into 14. So you are having data points in thousands, literally in thousands. So next application is, so somebody is not interested in counting the T cells and B cells. The person is interested in, person is working on cancer cells, and he wants to test whether his drug or his molecule, his herbal extract or his extract or whatever, nanoparticle, is killing the cancer cell or not, right? So can we detect the live and dead cells in flow cytometer? Yes, we can do that. So here what we are doing, we are again taking the cells which you have treated, and we are adding a dye which is called as propidium iodide, which gives you a red color fluorescence. And propidium iodide binds to DNA. But the thing is, the propidium iodide can enter inside only those cells which are dead. So what happens, the live cells can pump out the propidium iodide. So I can say that in my mixture of cells, if 50% are dead and 50% are live, then 50% will be having binding of propidium iodide with their DNA, and 50% will be negative for propidium iodide. So I can say 50% cells will be giving me a red fluorescence, and 50% will be giving me no fluorescence. So here we will be mixing. Within seconds, we can put the tube into flow cytometer and tell the flow cytometer, please measure what? Size, granularity, and fluorescence. So you can see that here again, we are having 2 populations here. So on x-axis, we are having the fluorescence given by propydium iodide. So these cells are positive for propydium iodide. So obviously they are dead cells. And these cells are negative for propydium iodide. So they are what? Live cells. So within seconds in a given sample, I can find out how many cells are dead, how many cells are live, and whether yeah, so I'm audible again, right?
Yes, yes. Please share your screen.
Just a minute, sir. We'll just make you co-host so that you can share your screen.
Okay.
Okay, done.
And so you can see the screen, right?
Yes.
Yes.
Okay. So I have discussed about the live and dead cell analysis, where you can easily check whether the given drug is killing the cells or not, or whether the given drug is somehow affecting the cell death. Or not, right? So we go to next application, which is we can further extend our analysis or further extend our knowledge of cell death caused by your drug or by your molecule, where we can analyze the cause of death. So generally, we want that if we are working on anti-cancer drugs or cytotoxic drugs by which we can kill target cells, we want that those cells should induce signaling-based death or apoptosis, that is programmed cell death. We don't want that our drugs are causing the cell death by some mechanical stress or by some other kind of necrosis or other kind of toxicity. So by analyzing the cell death, we can find out whether the drug is killing the cells or not. And secondly, by this application, we can find out whether the cells are dying because of apoptosis or not. Right. Now, how to achieve that? So I just go with basic fundamental, as I always tell the people that before starting the experiment, before designing the experiment, it is very important to analyze the biology behind the experiment. So the biology says that a live cell is having its membrane intact, and in the membrane we are having a specific phospholipid which is called as phosphatidyl serine. So this phosphatidyl serine is generally present inside the cell, or you can say inner leaflet of the membrane. Now, if the cell is undergoing the programmed cell death or the apoptosis, then the phosphatidyl serine is getting flipped out. It can come out. So this phosphatidyl serine can be recognized by a protein which is called as annexin 5. And this annexin 5 you can label with any fluorescence, and it can bind to that phosphatidyl serine. And if the cells are dying, then we already know from the last experiment that propydium iodide will enter inside the cell, will bind to the DNA, and will be giving you what? Red fluorescence. So imagine I have taken 1 million cells and I have treated them with an anti-cancer drug for 24 hours or 48 hours. And after 2 days, I want to check how many cells are doing apoptosis or how many cells are dying because of signaling-based death or apoptosis. Now, in that mixture after the treatment, there can be 3 types of cells. First, live cells. Second, the cells which are currently doing apoptosis. And third, the cells which are already dead because of apoptosis. So cells which will be live, their phosphatidyl serine will be inside, so they will not be bound by annexin, as well as the PI will also not enter inside that. So all the live cells will be annexin negative and PI negative. The cells which are currently doing apoptosis, the phosphatidyl serine will come out. So it will be annexin positive, but still they are live, so they will be PI negative. And finally, the cells which are already dead because of apoptosis, they will be double positive. They will be positive for annexin as well as they will be positive for what? PI. So we take the cells, we add our stains, annexin 5 and PI, we put the test tube into flow cytometer, and we just run the instrument. Instrument will be giving you 3 parameters again: size, granularity, and fluorescence. So here you can see the graph. On graph x-axis we are having fluorescence of annexin, and on y-axis we are having fluorescence of PI. Again, we are making a quadrant graph. So in quadrant graph, what is happening, you can see that these are the cells which are negative for x-axis, negative for y-axis. It means they are negative for annexin as well as they are negative for what? PI. So I can say they are live cells. Here, they are positive for annexin, but they are negative for what? PI. So you can say that cells are now doing a process, but not completely dead because they are negative for the fluorescence of propydium iodide. These are the cells which are double positive, means positive for annexin as well as positive PI. So they are already dead, and the reason of death was what? Apoptosis. So you can see that most of the cells are live in this experiment. Some cells are doing apoptosis and some cells are already dead. If I treat them with the drug, you can see that now the live cell percentage is decreasing from 96. Cells start doing apoptosis as well as they are dying. And finally, increase the concentration of drug to higher level, then you can see that a hell lot of number of cells which are doing apoptosis as well as they are dying. So we can check the particular cell population, T cell, B cells. We can check whether cells are dying or not. And we can check whether the cells are dying because of signaling based death or programmed cell death, or they're dying because of what? Apoptosis. Now, the next application is again very important. So why this application is important? Because it is working on very interesting principle. It is working on very interesting phenomenon. So you must have heard about one drug, paclitaxel and taxol derivatives. So in pharmacy, they must teach, in medicinal chemistry and in pharmacology, that paclitaxel is a drug which can bind to tubulin monomers, and they block the synthesis of spindle fibers during cell cycle. So they block the cell cycle, or they block the cell division. And if the cell division is getting blocked, then it can be utilized as a very nice anti-cancer drug, because we know that cancer means uncontrolled cell division. So how we can prove that a particular drug is arresting the cell division or stopping the cell division of your cells? It is very simple. We know that in cell cycle there are 4 distinct phases. G1, growth. S phase is DNA synthesis. G2 is again growth and preparation for mitosis, and then M phase. But I can divide them according to DNA content if we are talking about human DNA. So in human cells, in G1 cells, the DNA content will be 46 DNA molecules. In G2 and M, it will be 92 because replication making them double. And in S phase, there will be between 46 to 92 because the replication is occurring. So I can divide the complete cell division based on DNA content into 3 parts. G1 phase, 46 DNA molecule. G2 phase, 92 DNA molecule. And S phase, between 46 and 92, it can be anything. So we take the cells, we fix them. Fix them means we don't allow to do further cell division, and we add the PI, propydium iodide. So propydium iodide will be binding to DNA. Now, when we put that tube into flow cytometer and make the graph, where on x-axis we are having fluorescence of propydium iodide, and on y-axis, for example, we are keeping the number of cells or cell count, then we get this kind of graph. It's very interesting because these are the cells which are having least DNA content, that is 46 DNA. It means these are the cells which are present in which phase? Z0 or G1 phase. These are the cells here on this peak are having highest amount of DNA. It means they are containing 92 DNA molecules in human, if we are talking about humans. So they are present in G2 and M phase. And these are the cells which are having DNA content between least and highest, that is between 46 and 92. I mean, these cells are what? present in S phase. So within given 10,000 cells, you can ask the software how many cells are present in this peak, means how many are in G1, how many are in S phase, and how many are in G2. So G0, S, and G2, G1, S, and G2. So the software can count exact number of cells that in your population, in your 10,000 cells or in your 50,000 cells, what is the percentage of cells present in G1 phase? What is the percentage of cells which are present in H phase? And what is the percentage of cells which is present in what? G2. Very interesting. And the whole experiment takes 45 minutes to do, including incubation, staining, and analysis. Now, nowadays we are computerized and we are having amazing software which can count the number. But I have heard a story and people said that initially with the flow cytometer, they were not counting softwares, or softwares were not able to count the percentage and count the number, but they were giving the printouts like this. So you know how the people were assuming the percentage of cells present in a specific phase. They were cutting these peaks and these areas with scissors, and they were weighing. So if the total weight of the complete peak is, for example, 1 mg, I'm just giving you a number, it will be less than that. Then in comparison to 1 mg, what is the weight of this? What is the weight of this? And what is the weight of this? So based on the ink utilized and based on the weight of paper, including that ink, because if you're having bigger peak, then there will be higher utilization of ink during the printing and it will be giving a slightly higher weight. Then here some weight will come and here some weight will come. And based on the weight comparison, they were calculating the ratio that how many cells are present in G1, how many cells are present in S phase, and how many cells are present in what? G2. Amazing thing. I mean, people have evolved in science with very basic things to very high quality software. Nowadays, it takes 1 second or 2 seconds to calculate the percentage. How many are in G0, G1, how many are in S phase, and how many are in G2M. Now, we look at the experiment where the cells are treated with control, DMSO. All of you know very well that DMSO is a control. So you are having the peak of G1, then you are having the peak of S, and then you are having the peak of G2. Right. Now, you are having a drug which you are using PD901. The drug is known to stop the cell cycle at G1 phase. So all the cells are stopped in G1. So you can see there is reduction of S phase as well as the G2 phase in comparison to control. And this is our drug paclitaxel, which is blocking the cells in G2 phase. So all the cells will pass through G1, they will pass through S, and they will be collected into G2. So if you are getting only the G2 peak, it means cells are getting arrested at G2 phase. If you are getting only G1, they are arrested at G1. And similarly, some other drug can block the S phase. So you can exactly find out whether your drug or whether your herbal extract is killing the cell. If not, then whether it is stopping the cell cycle. If yes, then in which phase of cell cycle. If it is killing, whether it is killing by necrosis, whether it is killing by apoptosis, whether it is killing by mechanical stress. So all the things you can actually quantify. And I personally believe that to get good results and to get amazing data out of some technology, data comes always in terms of absolute number. Quantifiable data is required. Because sometimes what happens if we are looking at the microscopy and if we are finding whether something is killing the cells or not, then you can make the system bias. System bias means you can focus your lens only in that area where you want to look. But if the software is doing the counting for you, then you cannot be biased about your own experiment. And you can analyze the data in the most quantifiable way possible. Here comes my next advice before going to next application. I hope you guys are having time.
Yes, sir, we are having time. You can continue.
Yeah, great. So the next advice is, I always tell that humans are very selfish. Selfish in terms of what? They look at a particular problem, or they always ask the question in a manner in which they themselves are getting some advantage. So I am looking at every problem or everything from my point of view. And what happens in the research, especially if you're looking, if you're doing biological research and if you're working with biological system, one should think from the perspective of the system itself. Because we know that being selfish is not a good idea even in the normal life, because whenever we become selfish, we start getting wrong things, we start getting bad things, right? So one should be unbiased and one should choose a perspective where the question is being asked from the point of view of the system itself. I give you one very simple example. If I say why the cell division is happening, right? So people will start giving answers. Cell division is required for the growth. Cell division is required for the repair. Cell division is required for the body formation. Now, if you look at closely, all these answers are selfish. Growth is happening for humans. Repair is happening for the human organs. Then body is forming for the humans. We are not caring at all that why the cell division is occurring. We are not asking the question to cell itself. But from the day when I start thinking about the perspective of the cell itself, then cell will say, I don't care about your growth, I don't care about your repair, I don't care about the body formation. I am doing cell division because whenever I eat, the volume of the cytoplasm increases, but the volume of nucleus is constant, and with time the nucleus is losing the control on the cytoplasm. So the best way to re-establish the control is to divide the cell into two parts, to decrease the volume of cytoplasm. So you can control. That is the answer of cell. And if we are having real answer, if we are actually thinking from the perspective of the system itself, then we start designing good experiment. Then we start thinking about designing good drugs and how to analyze them, how to analyze their effect and all. So lactic acid is making, lactic acid bacteria is making lactic acid, some other is making ethanol, some other is making this, they are doing fermentation, and we think that they are doing fermentation for us. And that is our selfish perspective. If you ask the bacteria itself why you are making the lactic acid, then bacteria will say, okay, if I convert the glucose into pyruvate, then all the NAD plus are finished. To recreate the NAD plus, I need additional reduction reaction where I'm converting the pyruvate into lactic acid. I get the NAD plus back and I can continue the glycolysis. So they are doing this thing for themselves. They are utilizing one extra reduction reaction so the system can go on. They don't care about our fermentation. Why plants are making starch if they cannot eat their own potato? So people say it is the storage food. Why they require the storage if they can do the photosynthesis continuously in the presence of light? There will be no condition where the light will be switched off, sun is always there. So they are not doing it for storage. They are continuously converting the glucose into starch to reduce the osmotic pressure, to prevent the osmotic pressure to burst the cells. That is their immune response. That is protection mechanism. That is their defense mechanism. So one should start thinking from the point of view of the system. Why proteins are folding? Because proteins are folded, proteins are functional. No. Functionality is the selfish approach. The real answer is protein will say, I'm having hydrophobic amino acid. I have to fold myself in a way that I can protect my hydrophobic brothers from the water of cytoplasm. Then we actually understand the entropy, the folding, the 3D structure of protein. And if we are actually able to understand delta G, entropy and 3D structure of protein, then only we can do molecular docking, drug designing. It is not like you just sit on the instrument and you start analyzing the data. No, you have to understand the biology behind that. And the best way to understand the biology is to change your corner. Stop thinking from your perspective and start thinking from the perspective of the system itself. And then some amazing experiments will come out. So let's go for a little bit complex application, that is immunophenotyping. So now I want to analyze within given blood sample, how many are T cells, how many are B cells, how many are macrophages, how many are monocytes. Can we do that? The answer is yes. You can take blood and you can take PBMCs. And in the PBMCs, you start adding the antibodies against different proteins. For example, CD3 present on T cell, CD4 present on T cell, CD8 present on cytotoxic T cell, CD19 present on B lymphocyte, CD25 present on regulatory or suppressor T cell, CD14 monocyte, CD33 monocyte, and so on. Why the word immunophenotyping is being utilized here? That by utilizing the antibody, you are phenotyping a particular cell. So nowadays, a cell is not explained by its function, but it is explained by the presence of protein, type of protein present on that particular cell. If somebody asks me, can you define the regulatory T cell or suppressor T cell? I will give the answer, yes, I can do that. Regulatory T cells are CD3 positive, CD4 positive, CD25 high, CD19 negative, OXP3 positive. So that is, positive means presence of protein and negative means absence of protein. So that is the phenotype, or that is the definition a cell for me. If somebody asks, can you explain helper T cell, TH17? Then I say, yes, they are CD4 positive, CD8 negative, they are positive for IL-17, they are positive for ROR gamma T. So that is the definition. Things are moving more and more based on protein-based characterization of the cells. So if you really are interested in learning these things, then you should start learning about phenotyping of cells. How we can define different type of cells in our body. Liver cells are having different set of proteins. Skin cells are having different set of proteins. Neurons are having different set of proteins. Immune system cells are having different set of proteins. Can we do that? Yes. And once you learn defining the cells based on their phenotype, then you are becoming advanced. Then you are doing so many new things, right? And what is the aim of our life? Why we have taken the biology? We want us to be different from average people, and we want to do something good in biology, whether it is business, research, and all. And for all these things, you need a good new perspective as well as good fundamental and good knowledge of the biological things. So we have taken the PBMCs and we have added these antibodies. Then we can analyze the data, and data is becoming more complex, but it's very simple. Based on forward scatter and based on side scatter, we are having three populations: lymphocyte, monocyte, granulocyte. Within lymphocyte, we can find out CD3 T cells, then CD19 B cells. Then within T cell, we can find out how many are CD4 positive helper cells, how many are CD8 positive cytotoxic cells. Then within CD4 T cells, we can find out how many are suppressor T cells, how many are non-suppressor T cells. Then within monocyte, we can find out how many are monocyte, how many are other type of cells. So more and more you start adding the antibodies, more and more you start increasing the proteins. More and more you can analyze different type of population in a given sample. I mean, if I take the bone marrow, I mean, so many of you are doing pharmacology experiments on rats and mice, and you just take out the bone out of it. You can isolate the bone marrow. And in bone marrow, you are having so many types of cells: long-term hematopoietic stem cells, short-term hematopoietic stem cells, common lymphoid progenitor, common myeloid progenitor. In bone marrow, you can get 17 to 18 types of cells. And if you want to analyze them, all of them simultaneously, then you find out what are their specific protein and start adding the antibodies. Imagine you are getting information about 10 types of population simultaneously from one sample, and in a day you are analyzing 7,200 samples. So at what level of research you can bring, what level of research you can bring into your domain in the field of drug discovery, effect of drugs, and how they are working and all. Now, the next application is cancer stem cell analysis. One of my favorite. So people who are interested in cancer biology, people who are interested in understanding the tumor. If I talk about a tumor, if I talk about cancer, then I will be saying it's a heterogeneous mass of cells. So in a tumor, all the cells are not identical. They are different from each other. And there are some cells which can start new cancer, which can start metastasis, which can be aggressive, which can be drug resistant. And we call those cells as stem cells or cancer stem cells. So what happens in the conventional cancer therapy, which generally getting utilized now, they kill all the cancer cells, but they cannot kill the cancer stem cells. And these cancer stem cells can create new tumor, and the tumor relapse will be very, very fast. But if we find out the drugs, if we find out the therapies, if we find out therapeutic regimens, which are able to kill only the cancer stem cells, then even if the remaining tumor is present, the tumor will regress automatically because we have killed the mother cells. So right now in the field of cancer biology, 80% of the labs working on what? Identifying, characterizing, and designing the therapies against what? Cancer stem cells. And we can analyze them by flow cytometry. So if anybody of you is working in the field of anti-cancer drug designing, they are having some herbal extract or some scaffolds or some molecules which they think they can kill the cancer cells, then we can work together to find out whether they can also kill the cancer stem cells. Because the molecules which are able to kill the cancer stem cells, they will be the real good anti-cancer drug targets. So how we characterize them? For example, we are taking the breast cancer tissue. Again, I am preparing the single cell suspension. Then I am staining them with the antibodies specific for which protein? CD24 and CD44, because I know that breast cancer stem cells are having low expression of CD24 and higher expression of CD44. So you add the antibodies, put them into flow cytometer, you get this result that these are FSC, you select the cells, then you look for the expression of CD24 and CD44. So CD24 low and CD44 high. So these are the cells which are actually what? Cancer stem cells. So in that given sample of breast cancer, 26.43% cells are what? Cancer stem cells. And we should target them. There is another marker which is called as aldehyde dehydrogenase enzyme, LDH, which also express highly
Nishant, sir, your voice is not audible. I think we have lost him again. He's sharing his screen.
anti-inflammatory molecules. Simultaneously, 12 molecules to 18 molecules we can analyze in one sample. That is equivalent to 18 ELISA. So 18 ELISA reactions per sample you can do in one day multiplied with 7,200. So that saves your time as well as that saves your cost. So even mRNA analysis, cytokine analysis, antigen analysis, nowadays shifting towards what? Cytometry. So now I will discuss the last application which I did in my lab, and I'm very happy about it. It is one of my favorite applications. Imagine you are working against Plasmodium. You have created anti-malarial drug. How you are going to test it? So what people do, they grow the malarial parasite of Plasmodium in a plate and they put the drug and they check whether the drug is killing the parasite, whether the drug is killing the Plasmodium or not. But I am against these kind of testing because you are killing the Plasmodium in a plate which is not its natural habitat or which is not its natural microenvironment. You should kill the Plasmodium in its natural habitat, which is the RBC. So you have to design a system where you can check whether your drug is killing the plasmodium inside the RBC or not. Can we do that? Answer is yes. So we have started with 300 molecules actually, which were said, it was the WHO project, which were said to kill the plasmodium in a culture plate. Now, what we have done, we have added propidium iodide on the RBCs, and we don't get any peak. RBCs will not give the propidium iodide signaling. Reason is very simple. They don't have DNA, and propidium iodide is known to bind to DNA. So there is no DNA. So propidium iodide is not binding. Now, if you infect with the RBCs with plasmodium, then add propidium iodide, it will start giving you a positive peak. Reason is very simple, because plasmodium is having the DNA. So the signal is not coming from the RBC. The signal is actually coming from the plasmodium, which is present inside the RBC. So you can say that 10.9% RBCs were infected with what? Propidium iodide. Infected with what? Plasmodium. 10.98. Then you start treating these RBCs with your drug. First drug, second drug, third drug. Out of 300, there was only one drug which actually killed the plasmodium inside the RBC, and you can see from 10.98%, the percentage decreased to what? 5.8%. So all 300 were actually killing the plasmodium in the culture plate. But out of 300, only one has killed inside the RBC, and that was the real good drug target. So even in the microbiology, we are doing the same thing. We grow the bacteria and we put anything and that is dying, and because of that you can say it is antimicrobial agent. It doesn't work like that. That is why most of the molecules which we find interesting initially, they are killing the cells. They failed in the further testing because they are not able to work in the natural microenvironment. I always tell the students that you grow the cancer cells in a culture plate and you put some gutter water in that, cells will die. I cannot say the gutter water is an anti-cancer agent. It should be killing in the natural microenvironment. It should be killing the cancer stem. So there are so many advanced tests which you have to do before getting excited to say whether your drug molecule is working or. So I hope I have given so many applications. There are so many other applications, like you can do the diagnosis of different blood cancers. You can even analyze the phytoplanktons because they have different type of fluorescence. People are using them into food safety nowadays. I have given one complete lecture where in Pune University, where I have explained how the flow cytometry can be utilized in microbiological applications. Then people are doing sperm quality analysis. People are measuring the mitochondrial potential with flow cytometry. People are understanding the cell signaling, DNA content, plant and cell analysis, circulatory tumor cell analysis. I always say about flow cytometry, what is the limit? Imagination is your limit. If you can imagine something, then obviously you can perform that by utilizing what? Flow cytometry. If you are having great imagination, then it is very easy to test that. So I will be giving the answers. Just give me a minute. You can contact me on my email, that is logicalbiology84@gmail.com. You can WhatsApp me 86986847792. You can subscribe my Logical Biology channel at YouTube. I have discussed so many topics in the same way with a different perspective. It's in the most logical way possible. We are having some very good online courses of theory, like advanced immunology you can learn, advanced pharmacology you can learn with us, advanced cancer biology you can learn with us, NMR spectroscopy, and many more things are coming. We are having a complete biology program where I am teaching all 22 biology streams every Saturday and Sunday. It's a 1.5 year program. Then we can work together in the field of research. We can work together in the field of science. And I'm always open for the student as well as for research discussion. You can contact me anytime. Thank you guys. Thank you very much. Now I would try to give answers of the questions. Right.
Thank you, sir, for a wonderful session.
Thank you, ma'am.
There are a few questions from the participant side. So I will,
Yeah, yeah.
on their behalf. The first question is, how can we use flow cytometry in neurological studies, especially for Parkinson's studies?
So when people are working on anti-Parkinson compound, then we can check the effect of these drugs on the membrane potential of glial cells and neuronal cells, and we can actually find out the presence of particular ion channels, particular neurotransmitter receptors on the cells after and before the treatment. And we can actually also try the flow cytometry in neurodegenerative diseases, where we can find out the potential of neuronal stem cells and their differentiation by flow cytometry. So there are so many people who are analyzing the chemical properties and the potential based properties of neurons with different kind of psychotic drugs, different kind of neurodegenerative therapeutic drugs, as well as different anti-Parkinson drugs. So it is possible to analyze.
Hello. I just want to request here those already have questions in chat box, like Professor.
Yeah, I can read the question. So Dr.
Is there. So I just request them directly. They can interact and ask the questions.
Yeah. So you can type the questions. I will read the question.
So Professor Giram, you can ask your question. You can unmute yourself and ask the question. Or I'll just unmute you. Just a minute. So you are unmuted. You can ask your question.
Hello, sir. Am I audible?
Yes.
Yeah. I can.
Sir, how to perform the cell sorting based on this flow data?
So the cell sorting can be done by utilizing two methods. One is fluorescence-based cell sorting, FACS. So there, what we can do, for example, I am just giving this example. So here in this graph, I am having PI negative cells, these cells, and the PI positive cells, these 5.8 to 4% cells. So I can, the flow cytometer sorter is a different instrument where I can give the direction to the software itself. Yeah, I want the cells to be separated in a test tube, which are present in this gate. So you just separate the cells into two test tubes, one which are fluorescently negative and second which are fluorescent positive. And the software, based on the emission and based on magnetic field of the cells, they can sort out the cells into two different test tubes. This is called a flow-based or fluorescence-based cell sorting, flow cytometry-based cell sorting. The other method is generally, which I utilize in my lab because that is a little bit cheaper. We do MAX, the magnetic activated cell sorting. So there what we do, if I take the PBMCs and if I want to isolate the CD4 positive T cells from the blood, then I use antibody against CD4 positive T cells, and those antibodies are labeled with a nickel bead. And then we put the cells into a magnetic field. All the positive cells are sticking to the wall and all negative cells are free flowing. So we throw the negative cells and we can collect the positive cells. So depending upon how much starting material you are having and how much cost friendly experimentation you want, you can utilize any cell sorting method, either MAX, magnetic cell sorting, or FACS, flow cytometry based cell sorting. And I can say that we have done very sensitive experiments where we have isolated the regulatory T cells. So from 50 ml blood, we get 5,000 cells. So from 50 ml blood, you get billions of cells, and from those cells, you are isolating 5,000. So that much sensitivity can be achieved by FACS or MAX. Yeah, so it is just giving the direction to the system that what cell you want to sort or what cell you want to take out from your population.
Okay, sir, thank you. One more question.
Yeah.
While working on T-ray, that is CD4, CD8, POXP3 and myeloid-derived suppressor cells, I am getting diffuse peak in the quadrant. So how to minimize this diffuse peak?
You have isolated the regulatory T cells by magnetic cell sorting or by FACS? Or you are just using all complete blood?
complete mass of that cell.
Yeah, so generally what happens with T-Rex, the percentage of regulatory T cell is, and when you take the complete PBMCs as initial material, you will always get diffuse peak. So what I will suggest, from max you separate out the CD4 positive, CD25 bright, and CD127 negative T-Rex. Then you will start getting sharp peaks. Then you will start getting better T-Rex. This I generally perform, and I get better resolution of the signal as well as good population to analyze. Otherwise, it is always 3 to 4%. Sometimes it is 1% also. Sometimes it is 0.5% also. And in the given complete sample, it is very difficult to identify.
Okay, sir. Thank you.
Yeah, CD4 positive, CD25 bright, and CD127 negative. These are the real T-Rex which you can analyze.
Okay, sir. Okay. Thank you.
Thank you, Dr. Giram. Thank you, sir. One more question from Dr. Deshkar ma'am. That please elaborate on applications of flow cytometry in cell permeation studies.
It is very simple, ma'am. If you really want to check whether your drug is entering inside the target cell or not, you can label your drug with FIT-C or with any fluorescent dye, and you put your on cells. And if it is entering inside the cell, then your cells will gradually become positive for that fluorescent dye. And for after adding the drug, you can select different time points like 15 minutes, 30 minutes, 1 hour, 2 hour, 4 hour, 8 hours. And at different time points, you can check whether your cells are becoming positive for the fluorescence or not, and how much positive. So with time, if the drug is actually entering inside the cell, then the number of positive cells and the brightness of fluorescent signal will increase. So it's a generalized practice where we can find out whether the cells are entering inside or not, whether the nanoparticles are entering inside or not. The required condition is you have to tag them with some kind of fluorescent tag. So generally people use FIT-C to label different type of drug, and we have done these studies in good number actually. We have done for so many people who are coming with their drugs to check whether the permeability is good or not.
Okay. Now Dr. Mahore has one question. Mahore ma'am.
So the question is, can we check the effect of substrate prebiotic on mixed microbial culture using flow cytometry? You are trying to say that you are having a mixed culture of microbes and you want to characterize them in the mixture, whether there is particular proportion or particular percentage is present or not. So yes, but for that we would like to have the information about what bacteria you are talking about and what is their DNA content. So we have to optimize the system, whether we can separate them based on their DNA content or we can separate them based on a particular. Right. So to characterize the mixture, it is a little bit tricky task because you need pre-information that which kind of bacteria can be there.
To just add, that how many microorganisms at a time we can add?
How many
organisms at a time we can test with the flow cytometer? Saying just an example, I want to
in a given mixture, in a given
sample.
Yeah, so that depends. I mean, generally nowadays people are utilizing flow cytometry for bacterial population analysis, and they are going up to 7 to 8 population analysis, as well as they are testing the sputum sample for the presence of Mycobacterium tuberculosis and other E. coli. So there they are testing 3 to 4. Bacteria you are looking for, we can test. But yeah, in microbiology, I think there are a lot of things which are still need to be optimized, because we are not having specific proteins or microbes like human cells. There are cell walls and they are nearly similar cell walls. Only 1 or 2 proteins are different, and how to use antibody against them. So yeah, many things should be optimized for microbiology. Right now, 10 populations are there or not, 3 populations are there or not. This kind of experimentation is going in microbiology.
A request to you, just stop the screen share and switch on your videos. Just my request.
You want to see me?
Yes, sir. We all the participants want to see.
So my internet connection is very bad. That is why the video is not coming. Otherwise, I would have started on second one.
So if I will stop the screen share and then I think it may work. Just
let me try.
yes, sir. Just we will try. Otherwise, we can off it.
Let me try. No, it is not working because I am not working on Wi-Fi. I am working on my hotspot.
Okay, sir.
Because electricity is not there. But yeah, you can see me on YouTube.
Okay, sir. No problem.
So, are there any questions?
Yes, Mr. Saras Tiwari wants to ask one question. Saras? Saras, can you please unmute yourself and ask question? Or otherwise I will... Okay.
Hello.
Yes.
Sir, I just want to one question that can we use any other intercalating agent on the place of propidium iodide?
Yeah, sure. There are so many dyes like Cytox Blue and 7AAD. You just require a proper fluorescence coming out of that dye, which is falling under the range of our lasers. So any kind of reagent or any kind of dye can be used. PI is used generally because it is very cheap. You can find 10 grams of PI in 5,000 rupees, where you can do 50,000 experiments. Yeah, so generally people are using PI and giving the examples again and again.
Okay, thank you, sir.
Any reagent. You just require the fluorescence coming out of it. That's it.
Okay, so next question is from Prachi Dugam. Prachi Dugam, she wants to ask one question. Prachi Dugam.
Hello.
Yes, Prachi.
Yes.
Hello.
Yeah,
so
Prachi.
Can we use this technique? Hello.
Yes.
Yeah, I can hear you.
Okay. Can we use this technique for solid tumors like brain cancer and to check whether they are metastatic or not?
So generally the final test of metastasis is you have to use animal models to inject the cells. But what we do to check the metastatic potential of a solid tumor. So if you are giving me a tumor, then I will create a single cell suspension again from that solid tumor. And that solid tumor single cell suspension, I will stain with metastasis specific markers, or you can say epithelial mesenchymal transition, EMT specific markers, and we can find out how many cancer stem cells are there, how many metastasis potential metastatic cells are there. So yes, many people are working using flow cytometry to identify metastatic potential Vala cells. Yeah, it is possible. So we are doing it in lung cancer and colon cancer in our lab, where we isolate the solid tumor and we analyze the cells. And in oral cancer, we have done one amazing thing. From the patient, we take out the oral cancer tumor, and from that tumor, we isolate the magnetically cell sorting we do for the T cells. And we are proving that the cells which are entering inside the tumor and cells which are present in the lymph node of the same person, they are having different level of exhaustion. Means they are getting tired once they are entering inside the tumor. That is where they are not giving you responses. And we are trying to reboot those cells. So yeah, solid cancer biology. So actually any kind of cell analysis you want to do, that flow cytometry is your destination.
Okay, sir. Thank you, sir.
Thank you, sir. Now I request Tanaji Nangude, sir, to conclude and propose vote of thanks. Tanaji, sir.
Yes, thank you, madam. Very good afternoon, sir. Actually, it was a really very informative session, and we are really happy to have you to share your experience and knowledge regarding the flow cytometry. Lots of doubts get cleared about the participant. You have started with the basic principles, You explain very well. To understand any technique, to learn anything, you need to design first and to go through it, take the experience and understand and learn. Basic, actually for any study, it is a very good basic to understand how to learn. You explain very well the functionality of the instruments, then you explain very well the interactions of cell with the light, then various experimental studies you explain with graphical presentations and explanations, which was really well to understand, easy to understand for the students as well as for us. Now, also you that motivated student to how to study and how to go for the research in this area, that how they can build their thinking to integrate this research. Also, you given some examples which are problem based, and you explain very well how to solve this problem, how to go through this problem to find out the solutions. And lastly, you explain all the application, including this about the cancer stem cell analysis. All the applications were very clear to understand, and the question answer session was also very energetic, that to get all the doubts clear. Really pleasure to have you for this session to understand very clearly about the flow cytometry, its basic principles as well as various applications. Where we got the new areas open for flow cytometry with your session. I'm thankful to you on behalf of Training and Placement Cell, on behalf of Dr. D.Y. Patil, Institute of Pharmaceutical Sciences and Research, Pimpri, Pune, for your valuable time for this session today. Also, I'm thankful to our management, our principal, for continuous support and motivation. I'm thankful to all TNP team to taking these efforts to organize. Also, I'm thankful to Dr. Jayashree Maure for introducing such a great speaker with that much of experience and knowledge in this area. And that was really great to all students to know the applications of flow cytometry more clearly. I'm thankful to all the participants for their time, also their participation, active participation, and I'm sorry for inconvenience happened due to the network issue meanwhile the session. So I'm thankful, sir, for your valuable time and guidance during the session, and hope we'll have a same kind of interactions in the future.
Thank you, sir. Thank you very much. It was good. Yeah, it was good to talk to people, and thank you for having the patience. That is the best part.
One more request I have. Please see the chat box for the feedback form. Fill the feedback. All the participants, please fill the feedback. Then you will get the e-certificate of participation of this webinar. Also, I request all, please start your videos so we can take one group photograph with the logical biology. Because we are not able to see you.
Sorry to interrupt, sir. Nishant, sir, can you please put on your video?
Yeah, that's why I said I'm working. I'm not having electricity and I'm working on my hotspot. So that's why 2 times there was network disconnection and I'm losing the voice and video again and again. Anyways, guys. Yeah. So once again, thanks. All the best, and let's work in the field of biology to do some advanced things and to make a team of students who are fundamentally so strong so they can fight with the people from USA, Europe, and other countries. So let's do something great in biology. Okay, guys. Thank you very much.
Thank you very much. Thank you, sir. So I'm going to end this session. Thank you all for your participation and patience.